get my laser pointer. So hi, everyone. My name is Niklas. And over the next 15 minutes, I'm going to show you some of the modeling work I've done over the last year to better understand changes in functional states of human brains. So unfortunately, healthy humans tend to be quite reluctant to let researchers stab their brains with electrified needles. But about 1924, this German scientist, Hans Berger, did something less invasive, and he just glued an electrode onto someone's head. But the results were just incredible. Instead of seeing meaningless, noisy signal, he saw these structured periodic oscillations, now known as the alpha rhythm. And even more miraculously, when the subjects were asked to do mental tasks, the EEG signal changed. So it was almost too good to be true. Can we quantify any brain state just by the presence or absence of various oscillations? And this is really the start of this project where we put this question to the test. Our collaborator, an anesthesiologist, recorded the EEG of his patients as he gave them an infusion of the drug Propofol. Propofol is a general anesthetic that's used in the operating room and causes people to lose consciousness. So what happens when consciousness is lost? What's changing in this EEG signal? And can we infer anything about what's happening in the brain? Now, if you're looking for oscillations in a signal, the first analysis you do, as I'm sure you all know, is a power spectrum. So we can take a segment of our EEG data, and we can use a Fourier transform to break up the EEG signal into each of its frequency components. And then when we plot the amplitude of each of these components, you can see the relative contribution of each frequency to the EEG signal. So when we look at the power spectrum of this segment, we notice that there's these two peaks. And these peaks represent neural rhythms, and we name them based on their frequencies. So we'd say that this EEG signal is containing an alpha rhythm and a delta rhythm. So we should be able to compute the power spectrum of our data and quantify the changes in neural rhythms associated with loss of consciousness. But when we do this, we didn't see what we expected. We saw this. So we don't see that there's a rhythm that's appearing or disappearing. Actually, the entire power spectrum is affected. It's as if it's rotating. So there's something aperiodic that's changing the signal. And interestingly, this isn't the first time that such changes in this spectral trend, as we call it, has been reported. In several recent years, several papers have correlated these broadband EEG features with different stages of sleep, following exposure to various other drugs, as well as memory tasks performed by older individuals, but not younger individuals. So this overall trend in the EEG, this noise component of the signal, can be used as a biomarker for various brain states, maybe even states of consciousness, but we don't really know what it means. And that's really what I'm going to be talking about today. How do we interpret these changes in EEG noise? What does it mean mechanistically for the brain? And to understand that, we decided to take a modeling approach. So what is the EEG anyways? Where does it come from? Well, if we zoom in on a little segment of brain, we might see something that looks like this. So you have all these various neurons, and they're going to be sending something called action potentials to communicate to other neurons. These action potentials travel along these cables called axons, and the other neurons will receive these inputs along these tree-like structures called dendrites. So there are these currents flowing through these dendrites and these axons, and these currents generate electric fields. And so every inhibitory synaptic event, every excitatory synaptic event, and every action potential will contribute in its own way to the EEG. So what exactly do these contributions look like? So a previous study has actually computed what the EEG response will look like in response to a single synaptic event. And I want to point out that the amplitude of this response depends on where the synapse is located along the neuron. All right, so we went and we did similar simulations for action potentials propagating along axons. What does an action potential do to the EEG? And here the story is a bit simpler because there's no scaling. You don't have to worry about the input location. A given axon will produce the same response every time it fires an action potential. So you can think of these responses as a sort of basis set for building the EEG signal up. And from here, it's actually really easy to calculate the EEG. So if you know what the electrical responses are, well, because electric fields sum linearly, all you have to do is sum up all of the contributions. So if you know every event happening in the brain, you can scale it by its location and orientation in space. And this is just due to electromagnetism, and you can compute the EEG. The only caveat here is, of course, that you need to know the location, the orientation, and the timing of every single event that's happening in the brain, which we don't. But despite this, we can see something that falls out just from the fact that this is linear, which is if all the events are happening periodically, if the timing of all of these events are happening in a periodic fashion, well, then the EEG signal should be periodic. And this is exactly what's happening when you see these EEG oscillations. A large network of neurons are all oscillating in synchrony, say every 100 milliseconds. So these electric events are happening every 10 hertz, and you see an alpha rhythm. So a lot of effort has gone into simulating neural circuits to understand why and how they start oscillating and at what frequencies. But we don't care about neural oscillations. We want to understand the noise. What's the noise doing in the EEG? And so we thought, well, okay, what if instead of these all happening synchronously and rhythmically, what if all these events are just happening completely randomly? What if they're all asynchronous? What would the EEG look like if everything in the brain was happening randomly? So to set up this model, we went through open source anatomical data. For example, it's loading. For example, the synapse distributions across dendrites on average throughout the brain, or the distribution of gray matter throughout the brain. And we found that the anatomy could actually be fairly simply parameterized in such a way that allowed us to calculate an equation for what the EEG spectrum should look like if all brain activity was uncorrelated. So basically, we treat the EEG signal as a stochastic process, and then we try to look at what that power spectrum should look like. And when you do this, we see this. So we found that the estimates from the model, parameterized by values taken from the literature, trying to count it up as anatomically accurately as possible, we see that's very similar to what we see experimentally. So here's just an example of an EEG spectrum of a human at rest that didn't display any obvious peaks. And for comparison, this is what the average spectra look like across patients. And in general, these patients did display rhythmic activity on their EEGs. You can see them by the peaks. But we still see that the overall spectral trend seems to be similar to that predicted by the model. Now, under the hood here, there's really nothing complicated going on. It's basically a linear model where the effort is in properly counting up the contributions of all the various different electrical events. What it tells us is something interesting, which is that completely asynchronous neural activity contains about the right amplitude and the right time scales to explain noise seen in the EEG. So from this, we concluded that random asynchronous network activity is a plausible source of EEG noise. I mean, there's a lot of parameters going on here, and we're really not assuming anything about the dynamics of the network. So the real question is, with this model, well, can we predict the effects of propofol? We saw the effect of propofol in the spectrum. What can we say about it? So the mechanism of action of propofol is actually well known. The drug targets proteins called GABA receptors. And the effect is that it slows the decay of inhibition back to zero. So whenever there's an inhibitory event, if there's propofol around, the inhibition hangs around for longer. Now, with more inhibition, one might expect that neural activity in general is suppressed, and this is exactly what is seen. Here's just some data from experiments in monkeys showing the average firing rate in areas of cortex and thalamus, different areas of the brain. And after they give these monkeys propofol, we see that the average firing rate in the brain decreases. So when we implement these two parameter changes in our model, we see that we have this rotation, this increase in low frequencies and this decrease in high frequencies. And the reason for this is actually pretty clear. If there's just less spiking activity in the brain overall, there's going to be less noise. But because we're increasing the decay time of these inhibitions, you're actually shifting some of the noise to lower frequencies. And so it actually increases the power at lower frequencies. And so we get this rotation. And if you remember, this is exactly what we saw experimentally, that we see this rotation similar to what's predicted from the model. So it not only predicts the resting EEG spectral trend, but it also predicts the effects of propofol on the EEG spectral trend. And this explanation doesn't require any differences in brain dynamics. It's just a physical consequence of the slower inactivation events and less overall activity. In other words, our conclusion is that this spectral steepening could just be an epiphenomenon of propofol's pharmacology. So we came to this conclusion. We have an explanation for the spectral trend, but maybe the spectral trend doesn't actually tell us anything about consciousness, right? we still want to know what's happening with consciousness. So our final question was, well, now that we have predictions for these effects, how can we remove them? So to motivate this, let's look at a toy model for a second. So here I'm simulating simply just a random spike train convolved with some exponential decay, and then I'm summing them together and I'm getting this signal. So mechanistically, it's very similar to what our actual EEG model is. And when all these events are completely random, we get a power spectrum with a well-defined analytical solution. But now, if these occurrences are varying periodically so that they're slightly more likely to happen at the peaks of these oscillations, the signal is going to be similar to this, but quite a lot noisier. If we look at the power spectrum, we see the same underlying trend with this peak superimposed. So this is how the noise interacts with these oscillations underlying the activity. And we took a very similar approach with our full EEG model. So we just went in and we fit our model for asynchronous activity in tandem with these little bump functions that represent peaks from neural oscillations. So here's an example fit before and after loss of consciousness. And using this method, we can estimate and remove any changes in neural noise. In other words, the blue here indicates power that can't be explained by asynchronous neural activity. And what we're really interested in is these blue components. So we can go through, fit our data empirically, and remove the estimated aperiodic components. So before I show you what this corrected analysis looks like, let me show you what the original uncorrected analysis would look like. So here I'm showing a spectrogram where we have gone through and computed the power spectrum over time. So we're looking at how the power in each frequency component changes over time. And at 0 here, this is when the patient's lost consciousness. So as I mentioned in the beginning, we can look at the power in the different frequencies and try to estimate changes in neural rhythms. But when we plot this over time with the raw EEG spectrum, we see that there's this increase in alpha power, and then after a delay, a slight increase in delta power, and then beta power goes back down, and it's not entirely clear what's going on. But based on our modeling results, we shouldn't be too surprised, because according to the model, Propofol is changing the structure of the underlying noise. So these spectral changes aren't just a result of neural rhythms, they're a result of other things. So if we go through and estimate the noise from asynchronous neural activity at each time point and remove it, we should get a cleaner estimate for these neural rhythms. And this is what we see. So on the left here, this is what the spectrogram looks like after we've gone through and removed all this asynchronous power. And when we look at these neural rhythms, we see quite a big difference between these two. We see now that following the infusion of Propofol, there's this increase in alpha and beta power, suggesting that Propofol is causing these rhythms, but that maybe they're not related to loss of consciousness. But we also see that the moment of loss of consciousness is all of a sudden very starkly related to this increase in delta power. So why the difference here? Well, as I said before, when you have Propofol, you're increasing noise at lower frequencies. And so this slow increase is actually being contributed by this increase in noise. So when we remove this, we actually get this much steeper effect, and we see that it's happening right at the moment of loss of consciousness. So we conclude that loss of consciousness is uniquely and precisely associated with a delta rhythm. And this is very interesting because delta rhythms are very famous for being markers of sleep depth, with the deepest stages of sleep being associated with the most delta rhythms. So our observation actually supports the idea that different states of consciousness are connected by similar underlying mechanisms. But more generally, what we're presenting here is a principled approach for removing noise in EEG caused by asynchronous neural activity. And we're hoping that this approach may be applicable for more general purposes by other researchers. So I'd be happy to take some questions if there's time, but that's the presentation.
Thank you very much, Niklas. That was fascinating. Time for a quick question or two.
Niklas, I was wondering, I noticed in some of your graphs, you didn't do the correction for 1/, frequency, I think, but for others you did. So there's a known signal in, I think, fMRI, where the signal decays at 1/f, 1 the frequency.
Yeah, so that's
which is responsible for the downwards trend you see on the earlier graphs, but then on the last one, you don't see that. So I'm guessing you correct for 1/f or not. So
that's what you're talking about?
Yeah, so this one, there's no trend down.
Yeah, so you're absolutely right. Since you're familiar with 1 over F, that's really what this study is trying to understand. There is this decay, but where is it coming from and why might it be changing? So in a sense, what we did is use, instead of just an empirical approach to fitting this line and removing it, using a more biophysically motivated model of this trend. And you can actually see,
we might be losing.
there's some available packages for just removing this trend.
Awesome.
Yeah, so what I was going to say is, just because there's this 1 over trend, it's maybe a mistake to just remove the empirical trend sometimes. Anyways, yeah.
Eric, a quick question.
Yeah, interesting project, but people have been recording EEGs from patients undergoing anesthesia for 50 years in some form. Is there a bigger data set out here to try these ideas? I mean, I know there's lots of work in this area.
Yeah, so there are other studies that have done this sort of analysis. A lot of them, I don't think any of them have actually published their data publicly. But there have been other studies that have shown this shift in the power spectrum that we see here during general anesthesia. But it comes back down to the question of, why is it changing and what's the best way to correct for it? So for example, there was a study just 2 years ago that showed that the slope of the power spectrum is correlated with loss of consciousness. So that's not really new. But nobody's actually gone in and asked why it's changing.
I think we're going to have to move on. Thank you very much. That was really interesting. And our next speaker is